to talk about the discovery of DNA, and we're going to do so kind of in a timeline, learning about how DNA was discovered through the lens of the scientists who discovered it. My goal for you is for you to describe the series of discoveries that has led to the understanding of DNA as we know it. So, we start out with we know, we want to know, right? Where are we in, I think this is the 1920s. So imagine it is 1920. This is what we know. This is what we want to know. So we have the ability to see, and we can look around and we see that kids look a lot like their parents, right? Or most of the time, kind of. They share similar qualities with their biological parents. We also know that there is a mathematical pattern to which traits show up where. And when we talk about genetics next semester, we will be able to understand and use those patterns. But we know there's a pattern to figure out which traits show up where. And we know that there has to be a physical something. Something is passed from parent to child that holds this information. That's what we know in 1928. What we want to know is what this molecule is. What is it that is passed from parent to offspring that holds all of this information? The two main contenders were DNA or proteins. Is it DNA, proteins, or something else that holds this information, that shows us and tells genetically what traits show up where? So, we're going to talk about the timeline of scientists to help figure it out. And the first one is Frederick Griffith. Frederick Griffith was studying bacteria. He wanted to know why bacteria make people sick. That was his goal. That was his question. So on that very first line, he wanted to know, why do bacteria make people sick? That's what he was studying. Why is it that someone gets infected with bacteria and they feel really, really yucky? So what he did was he studied the effect of two different types of bacteria on mice. He had two different strains of bacteria, two different versions of bacteria. They were one of those strains gave the mice pneumonia, and one of them didn't. So he named them based on what they looked like. There was the S strain, which caused pneumonia, and it grew in smooth colonies. So it looked really smooth when he grew it on a petri dish. So he called it the S strain for smooth. And it caused pneumonia. So where it says S strain, which caused pneumonia and grew in smooth colonies. Pneumonia is right here. It starts with a P. And he also had the R strain. The R strain is harmless. It does nothing. There are so many harmless bacteria living on and in your body right now. So it's just like adding to that. Or for mice. So these were his two kind of his two bacteria strains that he used to set up his experiment. Yes. No, it can keep us healthy. A lot of them just do nothing. They're just like living to live. But this is specifically in mice. A very similar thing happens with humans. There are bacteria that cause pneumonia, some that don't. Yes. Mm-mm. There are actually a lot of bacteria that like live in our digestive systems and help us digest things. That's why if you ever take antibiotics, sometimes your digestive system messes up a little bit because the antibiotics kind of kill some of those, but they eventually build back up. Okay, so this is his experiment. So you have little lines underneath each of your experiments. So you're going to write the results of each experiment. So we've got two kind of control experiments. We've got injecting a mouse with the S bacteria, and if they did this, the mouse died. Right? It got pneumonia. Mouse died. If he injected the mice with R bacteria, the mice were fine, right? Because the R bacteria is harmless. But then he took the dangerous bacteria and he exposed it to heat and it died. So he killed the bacteria and then he injected that killed bacteria into the mice and the mouse lived. So if the harmful bad bacteria is killed before it's introduced to the mice, then the mice are fine, right? Because it's just dead bacteria. But then he did something interesting. He took that safe bacteria here, the safe bacteria here, and he took the safe bacteria, the R bacteria, the safe dead bacteria, and the safe live bacteria. He mixed them together, and the mouse died. So something happened when he mixed these two safe ones, and it still killed the mouse. The mouse still got pneumonia. Why do you think that this mouse died? Okay. Not necessarily. There are a lot of bacteria that grow side by side. So we've got our heat killed S bacteria. Those are going to stay dead no matter what. We can't revive those dead bacteria. And we've got the live R bacteria. Anybody else have a hypothesis as to what they think happened and why this mouse died? It's just what you think. There's no wrong answer. Yeah, Jude. Too much bacteria? Put too much stuff in there. Anybody else? Frederick Griffith, his idea was that something in the dead bacteria transformed the safe bacteria into our bacteria. Something transformed the safe bacteria into the harmful bacteria. And he called it the transforming principle. And we know now that bacteria can take up genetic information from the surroundings and then use it. Not always, but sometimes. So, he has this transforming principle. This means that something transformed the S bacteria into R bacteria, or R bacteria into S bacteria. That's what happened. Something took those really safe R bacteria and transformed them into harmful bacteria. That's that line. Something transformed the R bacteria into S bacteria. And now we want to know what that is. And it took a lot of time to figure it out. That last Frederick Griffith, he was doing this stuff in 1928. Our next leap is to 1944. So a little over 15 years later, we've got Oswald Avery, and he used the same bacteria that Frederick Griffith did, but he did something else with them. He used, like I said, the same bacteria as Griffith, and he used 3 tests to figure out what the transforming principle was. So he used the same cells as Griffith. That's that first line on the second page. He used the same bacteria as Griffith, and he wanted to know what the transforming principle was. He was actually able to isolate it, to not just take dead cells, but to get the specific part of the cell that was the transforming principle. They had it. They knew what the transforming principle physically was. They had some in a vial, but they didn't know what it was that was in that vial. So they did 3 tests to find out what it is. What is this transforming principle? So I've got 1, 2, and 3, those 3 lines. Those are going to be our 3 tests. The first test is a chemical analysis. They went in there, they took that transforming principle, and they wanted to know the percent of nitrogen and the percent of phosphorus in both of in the transforming principle. And these proportions matched DNA's exactly. So they were able to see that they had 4 different samples of this transforming principle. And every single time they did this experiment, they measured how much, what percent of it was nitrogen, what percent of it was phosphorus, and that ratio. So that this number divided by this number, the first number divided by the second number. And they all came out around the same. We know DNA's numbers. We know what DNA is. We just don't know what it does yet. So we know that DNA sits around 15.2% nitrogen, and all of these are sitting around 15. We know that DNA is 9% phosphorus, and all of these are sitting really close to 9% phosphorus. And we've got this ratio for all of these that is sitting around 1.6, 1.7. So based off of this information, do we think that the transforming principle, this thing that was transforming our safe bacteria into harmful bacteria, do we think it's DNA or proteins? Proteins, even though our proportions match DNA? You are thinking exactly like the scientific community is thinking. This data is saying, hey, we think the transforming principle is DNA. And everybody else is like, do you know how simple DNA is? DNA is a very simple molecule. There's no way that all of the information to make a human is in a simple DNA molecule. It has to be proteins. Proteins are way too complicated for that. So even though this first experiment said, hey, the transforming principle might be DNA, they had to keep going. They had to keep working to prove it. So they did an enzyme test. They took enzymes and they added them to our cell extracts and observed whether or not they were transformed. So for 2, it's enzyme tests. And then Oswald added enzymes to the cell extracts to tell whether or not they were transformed. And I've got this chart in your notes as well. So what they did was they took enzymes that break down proteins. So they took the extract and they put an enzyme in it that broke down proteins. So once that's in there, that means there should be no proteins left in the mixture, right? If all of them are broken down. They took that enzyme that breaks down proteins, put it in the transforming principle, and then injected that, and our cells were still transformed. So whatever was doing the transforming couldn't be a protein because all the proteins were broken down. So they're like, okay, what if we do it with RNA? What if we put an enzyme in our transforming principle that broke down all of our RNA? And they did that, and they said, and it still transformed. So even though there was no RNA in the mixture, it's still transformed. So RNA can't be the transforming principle. And then they took an enzyme that broke down DNA. So they injected that enzyme into our transforming principle, mixed it around, so there's no DNA left in that sample, and they put it in the culture, and our cells were not transformed. Which means that DNA had a key role in transforming things. Do you think this was enough to convince people that DNA was the genetic material? Are you convinced? Scientific community wasn't. We had to do another test. This one was relatively simple. They just looked at what was inside of it. What was it? Was there proteins in that in the extract? No. Was there DNA in the transforming principle? Yep. Did the scientific community believe them? No. So we still have to keep working to prove this, right? Scientists need a lot of proof before they can believe something this big. Because understanding what genetic information was a really, really, really big deal. So, we have some proof, we have some support for DNA being the genetic material. So, because of this, because we know that DNA is getting more important, we are having people spend a lot of time studying DNA. Specifically, a guy named Erwin Chargaff. So, he took DNA and he was looking at it, and he found 4 big ideas. So, we're in 1950, down at the bottom of the second page. 1950, Erwin Chargaff's base pairing rules. He always found the same 4 nitrogenous bases. Those bases are called adenine, thymine, guanine, and cytosine. Those are going to pop up a lot. So get used to those words coming around. And he was measuring the proportions of these bases. How much of the DNA was adenine, how much of it was thymine, how much of it was guanine, how much of it was cytosine. Cytosine. There we go, cytosine. Whew. Tripping over my words. He found that the proportions differed in different organisms. That means that the amount of cytosine in our DNA is different from the amount of cytosine in a banana's DNA. The amount of guanine in our DNA differed from a porcupine's DNA. The amount of adenine in spinach's DNA is a lot different than the amount of adenine in a cat's DNA. But here's the kicker. The proportions of adenine to thymine were always the same, and the proportions of guanine to cytosine were always the same. That's the top of your third page. I don't want that to blend in. I want you to put some stars around that. Up at the very top of the third page, where it says the proportions of adenine to thymine were always the same, and the proportions of guanine to cytosine were always the same, stars, exclamation points. Big idea right there. That is going to come in handy tomorrow. And then I've got a picture of all of those bases. This is what thymine looks like. This is what cytosine looks like. This is what adenine looks like. That's what guanine looks like. Just kind of wanted to have those available for you. Doing okay? All right. So now we get to 1952. We've got Alfred Hershey and Martha Chase. They are studying DNA. They want to make sure that DNA is the transforming principle. They are out to do the same thing that Oswald Avery did. Figure out, is DNA genetic material? Is it DNA or is it proteins? Because at this point in the 1950s, we were pretty sure that it was either DNA or proteins. It had to be one of the two of them. So, what they did was they studied special viruses that affected bacteria. So, just like there are viruses that affect us, there are certain viruses that only affect bacteria. And we call them bacteriophages. Now, bacteriophages and most viruses are very, very simple. All they are is DNA with a protein codon. That's it. Viruses are just DNA and proteins. Some of them are RNA and proteins, but we'll get into that a little bit later. Viruses, just DNA and proteins. Very, very simple. So, they are perfect for us to figure out which one it is. Is it DNA or proteins? Because viruses are just both of those. There's no other interference. And there's a key difference between DNA and proteins that was the key to their experiments. And that was their chemical composition. Proteins contained sulfur, but no phosphorus. And DNA contained phosphorus, but no sulfur. So what they were able to do is they were able to radioactively tag phosphorus in DNA, and they were able to radioactively tag the sulfur in proteins. And they were able to track where these molecules went when these viruses infected bacteria. Doing okay? All right, so I've got a little diagram there at the bottom for you. Here's what happened. So, experiment number 1, right up here. What they did is they took a virus and they radioactively labeled the proteins. Right? So they took it and they said, okay, the proteins in this virus are radioactive. So what they did was they took these viruses and they took some bacteria and they mixed them together. They let the viruses infect the bacteria. And then what they did was they literally put them in a blender. Like ran down to the store, picked up a blender, used it in the lab, put them in a blender. The goal of putting them in a blender was to then shake the viruses off and just get the cells. So they took what was left in this blender after they spun it around, and they put it into a test tube. And then they took this test tube and they spun it in a centrifuge. And what doing that does is it makes sure all of the heavy stuff sinks to the bottom and all of the viruses and the light stuff is on top. So we know down here in the bottom, that's just our bacteria. And then they were able to take that bacteria and look at it. And they were able to see that there was no sulfur in our cells. Make sure you write that down in that little space next to your experiment 1. If there was no sulfur, because there's no radioactivity here, it's not radioactive, so there's no sulfur, that means that no proteins entered this bacteria. This means that no proteins entered the bacteria. So, okay, we don't think that this genetic material is proteins. So what is it? I'll try it again with DNA. What they did was they took one of these proteins, one of these viruses, They took one of these viruses, and instead of putting a radioactive coat on it, they just put radioactive DNA in it. So they took DNA with this radioactive phosphorus in it. So it's in here. You see how it's super light green here? in the DNA this time? Because that's where the radioactivity is. It's in the DNA. And they did the exact same process. They put the viruses and the bacteria in the blender. Cleaned blender. Cleaned it all out. Spun it down. Made sure everything was all separate. Put what was ever in the blender into a test tube. Got just the cells back. And when they looked at those cells, they found the phosphorus. Now, these cells have that radioactive phosphorus inside of them. What does this tell us? What does it mean that there's phosphorus in these cells but no sulfur? Yeah. That it's DNA. What's DNA? All right. Our viruses are injecting their DNA into these cells. This is the experiment that told us, hey, we're almost 99.99999% sure that DNA is the transforming principle. DNA is what we're looking for. So, this is what leads us into DNA structure, which we'll talk about tomorrow. So, now, where's my clicker? I set it down because I always set it down. So, this is what we know. Last little page. A summary of what we've talked about today. We know for sure DNA is the genetic material. Thanks to Oswald Avery and Alfred Hershey and Martha Chase. Now, Alfred Hershey won the Nobel Prize for this and did not credit Martha Chase, but we will talk about women getting snubbed in science tomorrow. So, we know DNA is the genetic material. Thanks to Oswald Avery, Alfred Hershey, and Martha Chase. We also know what DNA is made of because of Erwin Chargaff, right? With the adenine, thymine, guanine, and cytosine, we know the proportions of those. We want to know how DNA works. How does DNA work? What does it do? DNA is so simple. How on earth is it the genetic material? How is all this stuff that is passed from parent to offspring carried in DNA? But in order to figure this out, in order to know how DNA works, we have to know what it's shaped like. Unlocking the shape of DNA was the key to unlocking the world of genetics, right? Understanding how DNA works is contingent upon knowing what shape DNA is. And that is what we're going to talk about tomorrow. Anybody have any questions?